Welcome. My name is Stephen Freeland. I'm the coordinator of the Environmental Speaker Series, brought to you by the College of the Environment in collaboration with the Salish Sea Institute and the Western Washington University Alumni Office, and today also in collaboration with the College of Science and Engineering, CSE. So a whole bunch of people helping bring today's guest to share her knowledge with you. Today, as always, I'm going to ask you to turn off those laptops, cell phones, put them away, ringers off, screen-free environment. Thank you very much. You don't need your laptop. Yeah, I know, really close them. With the exception of the TA in the back and me trying to keep the Zoom audience happy as well. And yes, welcome to the Zoom audience. We'll have time for questions at the end, either from the room or from the Zoom, and we'll get to as many as we can. Our guest today is Deb Donovan. Deb's over in the biology department, working with marine invertebrates. We'll hear a bunch more about that. If you've been paying careful attention this quarter, I think at least 2, maybe even 3 of our speakers have referenced Deb and her work. So it's perfect to have her in person so we can hear it directly from Deb Donovan. Welcome.
Thank you. I'm curious who mentioned me.
We can get into that later.
Hi, everybody. Thanks for being here, and thanks for having me here. Before I get started, I want to just tell you a little bit about how I came to be standing here. So I did my PhD up at the University of British Columbia, and actually was living in Bellingham and commuted up every day for 5 long years. But I did my PhD on abalone. My PhD advisor was open to whatever kinds of questions I wanted to answer, but I had to work on abalone. And so I said, okay. And so I did my dissertation on the energetics of locomotion in abalone. And when I got hired at Western, I started, I wanted to continue working on abalone, but for reasons that I'm going to explain, I was unable to get permits to collect them. And so I kind of shifted my focus to working on different kinds of invertebrates, mostly snails and sea slugs and scallops and stuff like that. And I did a lot of work on how they move, how much energy they use, how they change when the environment changes. And then I realized that, I might find scallop swimming fascinating, but it's not really going to make a huge difference to anything. At that point in time, so about 5 years ago, I had a graduate student who was working for the Puget Sound Restoration Fund. And I'll tell you more about that group in just a moment. But they run an abalone hatchery, and they're really working on restoring abalone to the Salish Sea. And so I decided then that I would work with them, that I would collaborate with them and kind of come full circle and work on abalone again. And I made that decision because I felt like the work that I could do and that my students could do would really benefit an iconic local species that is endangered. And so basically for the rest of my career here, I plan to be working on different aspects of abalone restoration. So with that, I'm going to talk about restoring Pinto Abalone in the San Juan Archipelago. Basically, what I'm going to do is talk about the work of my graduate students, and I actually have a couple of them right here. So you'll be seeing some of their preliminary results today. So I'm going to go through 5 different kind of small projects. So one from each of the grad students listed on this slide. I'm going to try to give you enough of an overview of how they did their work so that you can understand the results. And I pulled the kind of the most interesting results from their theses. The theses themselves are much more, they entail a much more information than I'm able to tell you about this today. Okay, so abalone. Abalone are large snails. The big ones in California can get to be this big. Our species, Haliotis kamtschatkana, or the Pinto Abalone, the largest ones get to be about this big. They are in severe decline around the world. And the reason for that One of the reasons for that is that they are very, very tasty. So people like to eat them. And there's actually farms, abalone farms around the world. We don't have any in Washington, but California, Australia, South Africa, all have abalone farms, New Zealand. And this is some prices of abalone from a California abalone farm. So I know it's small, but the actual meat of the abalone is going for $125 a pound. Okay, a live abalone is 40 bucks. And these aren't even really big ones. They're pretty small ones. Okay, so they've been drastically overfished around the world because people pay a lot of money for them, right? The other thing is that they're prized for their shells. So the inside of their shell is very pretty. You can make earrings out of them. And so they've been collected for their shells as well. So because of these pressures, abalone populations have plummeted in most parts of the world. Another important thing about abalone is that they are what we call ecosystem engineers. So they are grazers. They have this rasped tongue, and I'll show you some pictures of that in a little while. But they have this radula that they use to scrape food off of the rocks. And in scraping that food off the rocks, they clear space. And that cleared space can be colonized by other animals and plants and algae. No plants, just algae. Algae and animals. So the point here, though, is that they change their surroundings in a beneficial way that increases community structure in the areas that they are. So they're important as a food source. They're also important to indigenous peoples. They have been collecting abalone for thousands of years as a food source. They're important for the ecosystem that we live, or that they live in. Okay, so Pinto abalone are the only abalone species in our neck of the woods. They range from Alaska down to Point Concepcion in California. There's a lot of abalone species in California, but as you get into Washington, British Columbia, and Alaska, they are the only species here. Okay, so this is our one abalone species. They also, because the populations have been declining so much, they used to have fisheries for abalone in Alaska and in Canada. They had commercial fisheries for Pinto abalone. Those fisheries have been closed. So in Alaska, recreational harvesting of abalone can still occur, but the fishery has been closed. Canada closed their fishery in 1990. Washington closed it in 1994. I was hired here in 98, which is why I could not collect abalone to work on. So I couldn't even get a permit to do science with them. And then below Washington and Oregon and California, there was never really a fishery for them, but all harvesting has been shut down. Okay. So you might think, oh, okay, we're not fishing them anymore. They're going to bounce back. It's going to be great. Unfortunately, that's not the case. So the Washington Fish and Wildlife, Department of Fish and Wildlife, in conjunction with Puget Sound Restoration Fund, I'm going to call them PSRF for the rest of this talk. So Fish and Wildlife and PSRF have been monitoring abalone populations at 10 different sites in the San Juan Islands, in the Salish Sea, San Juan Archipelago. They started in 1990. And you can see that despite the fishery closure, abalone populations have continued to decline. And this point here represents 12 abalone at all 10 sites, a total of 12. So clearly they are not rebounding. The reason for that, well, there's probably a lot of reasons. One of the main reasons for that is that abalone are broadcast spawners. So they put their eggs and their sperm out in the water when they are reproducing. Hopefully egg finds sperm, gets fertilized, larval abalone is produced, and then the larvae swim in the water for about 7 days. They settle down to the bottom of the sea, well, the benthos, and they metamorphose into juvenile abalone, and then they grow up. So if you are a big snail and you need to find mates, if there's an abalone over there and an abalone over there, the chance of successful fertilization is pretty much zero. So what abalone do is they aggregate into spawning groups. They spawn synchronously. So if there's a very low density of abalone, those aggregating groups aren't going to happen, okay? So at this moment, there is very little, if any at all, natural reproduction of abalone in the Salish Sea. I'm going to also point out that this number here, so this y-axis is the density of abalone, and abalone per meter squared. It maxes out at 0.2. It's estimated that there have to be at least 0.3 abalone per meter squared for successful reproduction to occur. So that's not even, so 0.3 is up here somewhere, right? So it's important that we increase, that we bolster the abalone populations. Okay, so as I mentioned, Puget Sound Restoration Fund monitors 10 sites. These sites are on this map right here. And so there's just sites throughout the San Juan Islands there. And the recovery goal is to reverse the decline of Pinto Abalone stocks and create self-sustaining populations. So that's the ultimate goal that Puget Sound Restoration Fund has. Okay, so the people in my lab group, that's what we do. We work on different aspects of abalone restoration. And there's different steps that can happen. So the PSRF has a hatchery out on the Olympic Peninsula. They have wild-caught broodstock that they've brought in to the hatchery. They spawn the broodstock, they fertilize the eggs, and then they grow up the abalone in big tanks until the abalone are ready to be released into the wild. That period of time between spawning and release could be anywhere from, well, I'll show you some data in a moment, but it's usually about 12 to 18 months. So that's a year to a year and a half of feeding, taking care of, and watching these animals grow. So that's expensive, okay? The second step in terms of bolstering populations is to maximize outplants. And by maximizing outplants, I mean successfully outplanting abalone, having them survive and grow up to reproductive age. And then finally, there's this continued monitoring of abalone populations in the Salish Sea. Okay, so I'm going to talk about some work that's being done by my grad students. And I'm going to first talk about my student Lillian, and then I'm going to work on, or tell you about Caitlin's work. They were both involved in this first step of the restoration process. So Lillian was interested in how we could best feed the, we call them post-set larvae or post-set abalone. So abalone go through a larval phase and then they settle down onto the benthos. And then for a period of like 3 or 4 months, we call them post-set, and then they kind of transition into a juvenile. So when abalone settle, the first thing that they start to feed on are diatoms, which are these usually kind of single cellular algae that float around. Well, they can encrust things, but they can also be in the water column. So PSRF feeds their growing abalone a series of diatoms. But the question was, what kind of diatom will best support survival and growth? Okay. So what Lillian did is she chose 6 different types of diatoms that are fairly common around here and that can live in our cooler waters. She put the diatoms into little 6-well plates, little containers, put the larval abalone in them, and then watched them for 2 months. Okay, and she kept the temperature constant. She had them on a light dark phase so that the diatoms could grow. They're photosynthesizers, so they need that light. And then she measured abalone survival and length after two months. So what did she find? First of all, she found that there was a particular diatom called Amphora salina that best supported survival. Okay, I want you to notice this one, this A. brevipes was no better than the control. So those poor guys were starving to death. That was a really bad diatom for them. You might wonder how unfed, so the control were unfed animals. You might wonder, well, how could they do that? How could they survive for 20 days without being fed? Turns out that abalone larvae have enough yolk left over from their egg to survive through settlement and through about 20 days of life, okay? So they can survive on that stored energy for quite a while. So these poor animals were surviving on their yolk, and then when the yolk ran out, the food that they had available was not appropriate, and they didn't make it. Okay, another diatom, Cylindrotheca clusterum, was best supported growth, and that's represented by this line right here. But the other ones were fairly good. We didn't get growth off of these guys because they were all dead. So the hatchery now uses a combination of these 2 diatoms in their hatchery as the food for their abalone, for their juveniles, or for their post set. And so that you know, this really informed what the hatchery is able to do. Okay, the other thing that Lillian worked on was the abalone radula. So this is a scanning electron micrograph of an abalone radula, and you can see that it's got all these teeth. They've got all these raspy edges, and so this is the apparatus that the abalone uses to scrape diatoms and algae off of the rocks. She was curious whether, if being fed different types of diatoms, would change the morphology of the radula. So she hypothesized that if they were eating tougher diatoms, the radula might become, I don't know, different in shape somehow compared to if they were eating diatoms that were less tough. So she did all these dissections and measurements and compared all the radula, and it turns out that no, the type of diatom makes absolutely no difference to the radula at all. So now the hatchery knows that you know, whatever diatom they feed is not going to change the morphology of the animals. Okay, so I'm now going to talk about Caitlin's project. Caitlin was interested in the best method to cryopreserve sperm, abalone sperm. And you might wonder, well, why preserve sperm? I mean, what good is that going to do? So basically, the bottom line is that it helps to improve genetic diversity of the outplanted abalone. And it also helps with spawning events. So spawning is not always 100% effective. So the way that spawning happens at the hatchery is the PSRF folks will take maybe 3 females and 3 males. They put them in buckets. They heat the water up a little bit. They add some hydrogen peroxide because that induces spawning in abalone, and then cross their fingers. So hopefully they all spawn, and then the eggs and sperm can be collected and used for making larvae. So what happens if 2 females spawn and no males? Well, we've just wasted all of the eggs that those 2 females produced, and they can't spawn very often. They have to wait for a while to regenerate those eggs. So by having preserved sperm that could be used to fertilize those eggs, that would increase the production of the hatchery. The other thing is that it's really important to maximize the genetic diversity of these animals. So as you can imagine, since there are so few abalone, the brood stock that PSRF keeps is not a huge number. And so these animals are being used continuously to produce these larvae and juveniles for outplanting. And so it's important to be able to as much as we can, outplant genetically diverse abalone. Okay, so having cryopreserved sperm would help with that as well. All right, so Caitlin, it's actually really difficult to take a living cell, freeze it, and then thaw it and have it still be alive. Okay, so one of the things that Caitlin first of all did was figure out, well, how am I going to know how healthy these sperm are? And so she used a tracking software that was used to quantify sperm mobility or motility. So she could actually videotape the sperm moving, feed that videotape into this tracking software, and then be able to quantify how active the sperm were. And she'd get these little traces, and then the software would do its magic and figure out the average motility of these sperm. So there's Caitlin. Caitlin lived for a summer in this very cold room. And she's working with her cryopreservation stuff there. So basically the first thing that she had to do is she had to figure out which cryopreservative and in which concentration is best. So there's a number of different molecules that you can put into with these cells as you freeze them. And those molecules act to, basically they act to protect the cell. They protect the cell membrane, they keep it from bursting, they keep the insides of the cell from being dismantled. So these chemicals are very important for this cryopreservation. So she figured out which cryopreservative and the concentration that was best, and she looked at the survival and motility of the sperm. Once she'd figured that out, then she could figure out the freezing rate and the thawing rate that was best. So you can't freeze something really quickly and have it survive. You have to kind of go through the stepwise freezing to keep those cells alive. And the same thing with thawing. There's a particular rate of thawing that's going to be best. And so she worked all of that out. Finally, after she kind of figured out her protocol, the ultimate question was, well, can I freeze these sperm, thaw them, and will they actually still fertilize an egg? And so that was kind of the waiting with bated breath to see if this was going to work. And yes, she was able to do it. She's still working out some of the finer points of her protocol. So this work hasn't been published yet, but hopefully at some point it will be. Okay, I'm now going to talk about 3 students' work who are working on maximizing outplants. So maximizing the survival and the growth of outplanted larvae. And I'm going to talk about Katie first. Katie's already graduated. Then I'm going to talk about Lizzie and Jacqueline's work. They're sitting right here. So they're there, and their work is in the preliminary. They're just analyzing results and getting their work done. Okay, so Katie was interested in whether larval abalone could be successfully outplanted. So the hatchery usually outplants juveniles, the 12 to 18-month-old abalone. But remember that it's expensive to raise those animals up. So if we could successfully plant larvae or outplant larvae that are only, say, 7 days old, that would save a lot of time, a lot of money, and it would also potentially be better for the survival, because animals that are raised in captivity don't have the normal response to predators that other animals would have. They might not have the appropriate feeding behaviors, things like that. So it's thought that if we could outplant them at a younger age, that would also help survival. So Katie outplanted larvae in at actually two sites right off of Anacortes. So this is where Shannon Point Marine Center is, the Marine Center associated with Western. So she outplanted in these two sites. This site didn't end up happening. And she also had larvae being held at Shannon Point. So what she did is she took larvae out into the field, and she had these, I'm going to just show you this picture. These are crates that she constructed that had settling plates on the inside. So she took these, she called them larval abalone modules, lambs, and she took these out into the field, and she, excuse me, she put them on a big line. And for 3 of the, so there'd be 6 of these lambs on a line. 3 of them, no, I'm sorry, there were 9 on a line. 3 of them had this tenting over them. So you can see the tenting on the, here's one of the lambs. This is Katie diving right here. That's the lamb there. And 3 of them had tenting. 3 of them were just open to the ocean. And 3 of them were the controls. So what she did is you can almost see it here. Oh yeah, you can. So she had these big syringes full of water with abalone sperm. I should have brought some water. I did not. I think I'll be okay. All right. Anyway, she brought these big syringes full of abalone sperm. She squirted them into these lambs, and then she came back, took the tenting off, and then checked every month for 4 months to see if there were juvenile abalone in these lambs. So her question was, can larval abalone be successfully outplanted? And, and does this tenting help? Okay, all right. So what did she find? Yes, yay, larval abalone can be outplanted, and the tenting definitely helps. So if you think about it, if you're squirting in all of these larval abalone, I said sperm earlier, didn't I? She was squirting in larval abalone into these lambs, and they had been, and they were ready to settle. If you have these small larvae that are swimming around, if the lamb is open, they could be swept out. So having that tenting around kept them in that lamb and increased settlement. Okay. So this graph here shows the abalone abundance. There's our controls that didn't have any larvae put into them. Those are the open lambs, and those were the tented lambs. So we know that this is a successful thing that can be done. The abalone abundance, though, is still pretty low. So we're still working on those techniques as well. Okay. Now I'm going to talk about Lizzie and Jacqueline's work. And oh, God, thank you. Thank you so much. Hang on just a sec. I haven't talked this much in a while.
With a mask.
With a mask on. Thank you. Okay, that's much better. I'm just going to leave that open. All right. So, and I'm going to talk about they're doing very related projects. So I'm going to describe their methods together, and then I'll show you some of their data that are just hot off the press. So the water in the Salish Sea is very variable. It varies in pH, it varies in temperature, it varies in oxygen levels, it varies in salinity, and it varies on a yearly basis, which you can see here. So these are data that were collected at observation points or observation posts. And basically, these buoys collect data on a daily basis for years, okay? So you can see here that this is sea surface temperature. You can see that the temperature is increasing and decreasing depending on the month. So it's warmer in the summer, cooler in the winter. You can also see that pH is changing as well. But you can also see that there's a lot of variation. So these parameters vary not only on a yearly basis, but on a weekly basis and on a daily basis. Okay. So one of the things we know about the outplanting of abalone is that the site that the outplants are occurring, some of them are really successful and some of them are not successful at all. And we don't know why. One of the thoughts is that it could be these environmental parameters. So Lizzie and Jacqueline are both looking at the effects of temperature fluctuation and pH on abalone's survival and growth. So they have what we call a fully crossed experimental design, where they have two temperature treatments. So one is a constant temperature, it's constant at 10 degrees through the whole experiment. And then the other one fluctuates, and it goes from 10 degrees up to 14 degrees, 1 degree per day, and then back down to 10, and then back up to 14, and then back down to 10. Okay? So they have those two temperature treatments, and they also were able to inject their experimental containers with different amounts of carbon dioxide. And the carbon dioxide reacts to change the pH in the water. So they had two carbon dioxide treatments, two temperature treatments, and then they were fully crossed. Okay, so what did they find? Oh, right. So Lizzie is working on post-set larvae, post-set abalone, and then Jacqueline is working on juveniles about a year old. Okay, so there's Lizzie. She's looking through the microscope. This is what she's looking at. That's a little post-set abalone. You can see its little eye spots here. There's one eye spot. Its little tentacles, its shell. This is its guts there. So what she found, or what she's finding right now, is that survival in constant temperatures. So I'll just orient you to this graph here. So this is % survival here. And then these are the 4 treatments. So this is the constant temperature at 1200 parts per thousand, parts per million carbon dioxide, constant temperature at 400, fluctuating at 1200, and fluctuating at 400 parts per thousand. So what she's found is that, first of all, survival in constant temperature, constant was higher than in fluctuating temperature, but that higher than fluctuating temperature was really driven by this treatment here. So PCO2, the amount of PCO2 affected survival at the fluctuating temperatures, but not at the constant temperature. Why 400 parts per thousand, parts per million in fluctuating was lower than 1200 is something we're still trying to figure out. So if anybody has any good ideas, we would love to hear them. Okay, Lizzie also found that that growth was affected by both temperature and pH. So the constant, the two constant treatments are here, the two fluctuating treatments are here, and you can see that the fluctuating temperature treatments supported higher growth. And that kind of makes sense because as the water is warmer, abalone are going to grow faster because they're small invertebrates. Okay, she also found that the PCO2 affected the animals. So there's the 1200, 1200, 400, 400. So growth was lower in higher PCO2, and higher PCO2 means lower pH. So as pH goes down, growth also went down. Lizzie's also dissecting out tiny little radula, but she didn't have, she's still working on that, so I don't have any data to show you yet. Okay, there's Jacqueline. She's doing respirometry on juvenile abalone. What she found is that abalone survival was significantly greater in constant temperature. So that fluctuating temperature was really hard on them. And the hatchery also finds that the hatchery is, they really try to make sure that their temperature is very constant because they know that fluctuating temperatures are going to be hard on the animals. She also found that there's a temperature carbon dioxide interaction such that the constant temperature, let's see, it's driven by this right here. So the constant, the oxygen consumption at constant and low carbon dioxide was significantly lower than the other three treatments. Okay, so what this tells us is that having a fluctuating temperature and high PCO2 is going to increase the metabolic rate of these animals. Okay, so back to, hopefully what their data are going to allow the hatchery to do is to choose sites that might be more constant in temperature or have less changes in pH being affected by, say, water runoff or other factors that would change pH. Okay, so again, our big plan in the lab is to aid Puget Sound Restoration Fund in increasing the populations of abalone. I'm going to end this with some good news. So the 10 sites that I showed you earlier are just the monitoring sites. The outplant sites are super secret. Nobody knows, well, clearly somebody knows where they are. I don't know where they are. Because it doesn't make any sense to put a bunch of abalone in the water where everybody knows where they are and then wait for them to grow and then people are just going to go and poach them. I mean, poaching is a problem. So the outplant sites are super secret. They were just monitored. Well, they've been monitored. And so they've been outplanting abalone into these sites for about 10 years now. And at some of these sites, the abalone abundance is well above that 0.3 abalone per meter squared that we need for successful reproduction. And the abalone at these outplant sites are now big enough to start reproducing on their own. So fingers are crossed that at least at these sites, there will be some natural reproduction occurring in the near future, if it's not already happening now. So, okay, so I thank you for your attention, and I'm happy to answer any questions that you have. I'm going to get a drink of, just a minute. Yeah.
The ECO2 temperatures
Uh-huh.
that were chosen for the tests, were they based on like extremes or like averages of sales?
They were actually based. Did you turn it off?
Yep.
Okay.
Try now. Yeah. Thanks.
They're actually based on these data. So the temperatures, this is sea surface temperature, so it's a little bit different than the temperature that the abalone would be at. But the 10 and 14 degrees are temperatures that the abalone would see. Okay, and then the PCO2, the amount of PCO2 is linked to specific pHs, and we chose those pHs to be reflected of the variation that abalone might see. Yeah, so they were very carefully chosen. Yeah.
With increasing pHs of the oceans, how would that affect them?
Yeah, so it's decreasing pH of the ocean. So the more carbon dioxide you have, the lower the pH. That's why we call it acidification. So we know that, okay, just to back up for a moment. So with decreasing pH, that makes calcium less available to animals. And if you're making a shell, calcium's important, right? So the lower the pH, the harder it is for animals to calcify shells, or like corals have the big calcium carbonate structures. So one thing, and I didn't put these pictures in, but Lizzie has actually documented shell differences, so shell abnormalities in the animals as well. So that's where pH comes into effect. Did I answer your question? Good. Okay. Yeah.
I'm just wondering if there's been like any thoughts on potentially trying to intentionally breed stock that are the survivors of the fluctuating pH levels and whatnot.
Oh. So the question was, I haven't been doing the questions. The question was, is there an effort to breed broodstock that are resistant to the fluctuating temperatures? And not that I know of. Yeah. I think we're just lucky to find the other thing too, actually, I'm going to back up. One of the things that PSRF is very worried about is using wild broodstock. So they have a lot of abalone at the hatchery that are the offspring of wild broodstock, and they have not used any of those for reproduction yet. They're thinking about it, but they're concerned about the genetic consequences. But that would be something, you know, they've got these animals that have maybe survived fluctuating temperatures or pH or whatever, and they might be used. That's a good idea. Yeah.
Are the spawning events of the abalone dependent on, or is there any like relation to environment, whether that be like pH or?
Yeah, so the question is whether spawning events, and are you talking about wild abalone? Yeah, are spawning events of wild abalone related to environmental factors? Absolutely. Yeah, so there's usually a temperature. Temperature is important. Daylight, the amount of daylight is likely important. So abalone spawn in like the spring through the summer. And then it's difficult to get them to spawn after that. Yeah.
I just follow up about the wild versus like not wild rootstock. Is there like kind of what I'm wondering what your opinion is about the cost benefit of I'm trying to figure out how to word this. So like losing genetic diversity if you don't introduce wild broodstock, is that any sort of concern?
Yeah, so the other thing that, one of the things that PSRF is also thinking about is actually getting abalone broodstock from other places. So British Columbia and Alaska have much larger populations than we do. But then you run into the problem of you're introducing genes of animals from a whole other population. And is that something we want to do?
Yeah, just kind of wondering if the consequences of inbreeding versus the consequences for bringing wild,
Yeah.
places.
Yeah, it's something that they spend a lot of time thinking about the best way to do it. And there's a new professor at Shannon Point who's actually doing some work on the genetics of the wild populations. His name is Jay Diamond. And he is actually, well, he's done a couple of different things, but he's actually taken samples from abalone at the different outplant sites. And he's looking to see what kind of genetic variability is out there. So he's testing that directly. And then he's able to actually take water samples and with a huge amount of computing power, figure out if abalone DNA are in those water samples. And I'm trying to think of what he just finished that. I think he was able to detect abalone DNA at the outplant site. So that is a way we could monitor genetic diversity as well. Yeah, you're welcome. Yeah.
I was curious for the research that had those results that has been implemented in sort of the outplanting strategies at the PRSF. How many times did those findings need to be replicated, or how many times did the experiment need to take place to kind of know that it was, yes, let's go ahead and implement those sort of adjustments?
Yeah.
Versus, well, we'll test it again to see if it happens again.
Yeah, that's a really good question. And the only one that has been kind of finished was Katie's with the outplanting the larvae. And PSRF had actually done a prior study trying to outplant larvae. They used slightly different methods. So basically what they did is they made these big rocky piles, and they used super soakers, lab equipment, right? Use super soakers to inject the larvae into these big rocky ballast things. They went back, it was either a year or 2 years later, and deconstructed the whole thing. I mean, they had, of course, more than one of them, found zero abalone at all. So that was a little disappointing. And that's what gave Katie the idea of using these mesh coverings to try to keep the larvae in the container. So, and she did get, she was successful. So PSRF has not, I don't think, tried to outplant larvae systematically yet. They're still working with the juveniles, but it's definitely something that they're thinking about. Yeah. Yeah.
What has the role of all of these outsites been on the subsequent species in the area that were successful? And how relevant has it been of abalone reintroduction to the outset of the other species? How does it affect the other?
Excellent question. I have no idea. Yeah. So the question was, how has the outplanted abalone with the increasing populations, how has that changed the other animals or organisms in that community? And so PSRF has done some videotaping of different outplant sites to see what might be preying on the abalone. They're worried if they've got these islands of abalone that it's going to draw all the predators to that one place, right? And so they've done some videotaping. I know that there's hours of videotape waiting to be analyzed, but that hasn't been done yet, as far as I know. The other thing too is that these are fairly small areas relative to the Salish Sea. And so I think in that, the bottom line is I don't know, but we could, hopefully find that out. That's a good potential question. Yeah.
Yeah, we have
Oh.
one question online. How much has poaching affected natural abalone numbers in the Salish Sea, and what can be done to reduce the impacts of poaching?
Yeah, that's a really good question. And so we think that the decline, the initial decline, was a combination of the fishery and poaching, illegal harvesting. And after the fishery was closed, there were still people harvesting abalone illegally. I was just looking at some of the status reports today to brush up on numbers and stuff, and they did have a picture of a boat that had been confiscated. And the boat's name was like Abalone something. I mean, it was like an illegal abalone boat named Abalone Harvester. I mean, it wasn't that blatant, but it was close. And I was like, wow, that's really interesting. But it's definitely a problem. I mean, enforcement's out there. But frankly, in the Salish Sea, there are so few abalone that I doubt the poachers are even finding them. Yeah.
Okay, great. Next week, our guest is Abe Lloyd and Food System Sustainability. Deb, thank you very much.
You're very welcome. Thank you.